But it's my pleasure to announce the next speaker, and that's Michael Metzker. So Michael is a researcher, innovator, entrepreneur, all in one. And he's also been instrumental in the development of next-gen sequencing very early on, back in 2002, I think, in your early company. Now he's at Redfold Biosciences, and he's going to talk to us today about sequencing and the developments there. Thank you.
Good morning. Thank you, Claudia, and I'd like to thank the organizers for inviting me to talk. Actually, my very first paper on reversible terminators was published in 1994, so I am naive to the field of tuberculosis. My background has been in technology development, in genomics, HIV transmission, and other things. And so what I would like to do is kind of tell you a little bit about Red Vault, which is a company I started several years ago just to focus on innovations of genomic technologies, looking for problems, as I'm hearing here in this meeting, and trying to come up with creative solutions that can really advance the field. Let's see, do I control this? Go to the right. Great. So I don't need to tell you all of this, but as preparing my slides, this is a major global health crisis. The numbers of individuals affected and dying each year is staggering, and the problems are getting worse, particularly with drug resistance. And so I think sequencing technologies can make an impact. They can move into the diagnostics field. It's only been a little over 10 years ago that the first next-generation sequencing technologies were being revealed. This was really pushed by the National Human Genome Research Institute that issued RFAs for the $100,000 genome and the $1,000 genome. And we saw 454 come out with technologies, AgentCore, Personal Genomics coming out with the solid system, and then the Illumina system. All three came out within a 2-year period. And really over that decade, Sanger sequencing, which was the gold standard, it sequenced the human genome, really has taken a backstage, but yet is still an important and useful technology. And I don't think we should discard it in light of next-generation sequencing, because next-generation sequencing can't do it all. In fact, in many cases, it's overkill. It's too much. It's too much data to sift through, and it's wasteful in resources. I think you all know that major advantages are just enormous amounts of data. But that data is not necessarily perfect, and I'll make points why I believe that. Regardless of that, next-generation sequencing is in clinical markets today. It's being sold as tests in non-invasive prenatal diagnostics. There are 4 companies competing now, selling tests that draw mom's blood and can determine the genotype of the fetus. And in cancer diagnostics, there's many companies producing gene panels to identify cancer mutations that can aid in personalized therapies. Just a little bit of the work that I've done is HIV forensics. And so this is a figure that was taken out of a paper we published a couple of years ago. This was a criminal trial where an individual was accused of transmitting HIV to numerous individuals. And we relied on Sanger sequencing and phylogenetic analysis to ask whether the viruses from each sample were more closely related than any database or any other control samples that we could find. But the study was designed to ask a second question. If we could show relatedness, could we then show a direction of transmission? And that's what this data shows here, is that one of the samples, which were blinded to us, we had no idea who the case samples were, was showing a relationship of multiple infections, a paraphyllylic relationship, which was helpful in identifying the defendant in this criminal case. And I think these types of phylogenetic studies can also be very impactful in trying to understand the molecular causes of drug resistance and tuberculosis. We've heard next-generation sequencing. It is a composite of technologies that continue to evolve and change. I'm just showing some of the commercial instruments right now, the Illumina systems, the HiSeqs, the MiSeqs, the Ion Torrent, the PacBio, and the MinION. The PacBio and the MinION are single molecule technologies that can deliver really long read lengths. And that's one of the challenges with Illumina sequencing right now, is that even for a small genome like TB, you can't de novo assemble it. You have to rely on mapping technologies to try to find those differences. Or you have to combine PacBio data or MinION data with short read data to try and stitch together what a genome might look like. And whereas aligning reads to a reference genome might find single nucleotide variants, they don't find any other large structural rearrangements, insertions, deletions, and things like that. So those are some of the challenges still with next-generation sequencing. So what you want is predicated on how well the technology will deliver what you want. These figures were taken from two pictures just to highlight that next-generation sequencing is still in a discovery phase. We're still understanding the genetic variation of the TB genomes. A paper published by Zhang in 2013 identified a number of new genes that previously were not suspected as being involved. So I think as a diagnostic test, we have to get past and find biomarkers, as we heard earlier today, before we can really start relying on what are we asking and can we accurately and reproducibly get that information. And as I was thinking about the title of my talk, which was about devices, I was really struggling to figure out how I could actually tell you that next-generation sequencing devices for personalized therapies can actually be done today. And it can't, and we've heard several speakers kind of echo that, because it requires a number of things that next-generation sequencing does not deliver. Yes, a MiSeq can sequence a genome in a couple of days, but the library prep, the sequencing, and the data analysis all together take a lot longer and are not always comprehensive and complete. Next generation sequencing is still expensive. If you want to do a 4 megabase genome, even at $1,000, that's pretty expensive. Typically what you would do is you would barcode samples and pool them together, but that adds a new level of complexity and just informatics of keeping track of all that data. Accuracy and completeness also are not quite where we are today. We are learning we're learning a lot about the data that comes from next generation sequencing, but we're also learning the deficiencies. And so I think it is good to get technologies out in the research arena, really vet them out, understand their deficiencies, and go from there. And I mentioned this before, one of the current drawbacks is using a reference genome for alignment. At Red Vault, we've been working on a technology to amplify large strands of DNA directly on flow cells. So I'm getting into the weeds just for a second, but on an Illumina flow cell, once you do the sample or the library preparation and you introduce it onto the flow cell, you amplify single molecules and make multiple copies. These are the clusters that you will see as little bright spots on a fluorescent image. You can only amplify really short fragments, and this is part of the reason why you have to map to identify sequence variants. And it's a major limitation in the technology. And we've identified a new strategy where we can take a 10 kb fragment, take the genome, fragment it into 10 kb chunks, and amplify it onto the flow cell so that you can move the same technology that Illumina sells now from a mapping technology now to a technology that can do de novo assemblies. And we're working on this in cancer, and I believe for TB this would be as equally valuable. The sample preparation pipelines are still complex. It requires multiple steps. It requires lots of sample input. And the real source, sputum, seems to still be somewhat problematic of getting good and reliable data. And so can we simplify the workflow? And that's what we're working on at Red Vault, is to figure out ways of using the starting material as the catalyst without a library production or preparation. I mean, if we can eliminate library preparation altogether and feed DNA directly onto the flow cells, that would simplify workflows tremendously. It simplifies the amount of technical expertise that large centers still use to sequence big, large genomes. And I believe this could be tremendously valuable in rapid detection of drug resistance and global surveillance. So just kind of thinking, you know, despite there are a lot of other methodologies that can identify variants in TB, I still think whole genome sequencing is a powerful tool. It provides comprehensive data types and data sets. The difficulties is it's still a very complicated technology. It has weaknesses. It requires highly technical personnel. And we've heard a little bit of moving it into improved patient care, and I'm not here to tell you anything about that because I understand there are lots of steps. But the idea is at the forefront of the technology development. I'd like to see this technology move in a way that can connect all the dots to help the patient. So with that, I thank you for your time.
All right, thank you very much, Michael. Maybe one quick question, clarification. Otherwise, I'm sure Michael would be available to answer questions in the lunch break. All right, and he also will be on the later panel, so if there's nothing coming up right now, I think.
Thanks.
Thank you, Michael. So we'll get to the next stage of the session. A very exciting overview of our technology platforms. This will be like quick fire. We have 3 minutes presentations each from 5 different representatives from cool new tools. And we'll have 1 or 2 very, very quick questions after each, but then afterwards bring them up for a panel discussion, as I said earlier, moderated by Madhu. So I'll ask, just going from the top, first Chris Cooney, the Director of Engineering from Aconi, to come on stage. Don't see him yet. Ah, here. Yes. Very good. So Chris will present the Aconi development. And yes, Chris, please stick to the 3 minutes, and then afterwards stay here.
Sorry, Claudia, it's Charles, the Dage. Oh. No problem.
All right, sorry.
No. Chris and I were here to trick you, so that was our goal.
I haven't met them in person, so apologies.
Yes. So I'm Chuck Dage, the CEO of Aconi. This can't go against my 3 minutes. So I'm used to 5-minute elevator pitches, but now TB makes it even harder for us and do it in 3 minutes. But thank you, Michael, for that talk, because I was feeling pretty blue about sequencing and taking over the future of diagnostics that we're working on, which is trying to make microarrays easier to use. And that's been our focus of our company. Shown here, our goal is to have a flexible diagnostic platform and a common manufacturing infrastructure for products which enable multiplexed pattern recognition of genetic, protein, metabolite, or small molecule markers associated with the host or pathogen. So we want to look at everything across the board. I call it my if you like, buzzwords, TB, companion diagnostic precision medicine approach. But we want to provide value to clinical trial, patient stratification, disease prediction and prevention, early diagnosis, not just looking for TB and drug resistance, but co-infections that could be associated with it, treatment options, looking at adverse drug reactions. These antibiotics are brutal, and you take them for a long time. Shouldn't we look to see if they're going to have an adverse drug reaction to them before we prescribe them. And also, in addition, looking for drug resistant. And then treatment progression, as everybody's talked about, drug levels. Can we monitor reduction in inflammation or oxidative stress, toxins, things like that to tell you if you're progressing. So we're the baby in the crowd. We are in the design and development phase of a phase gate. We've dabbled in clinical, but we do not have a product launch, so I have to show you the potential and the pieces of our technology. What is at the core of our technology is the true array, gel drop array. And it's a large pore hydrogel that's mostly 99% water. And it's really faithful to aqueous-based reactions, which for those of us who are assay developers, when you do something in a tube and you try to put it on a surface as a monolayer, it doesn't do what you want. So these hydrogels come in really handy because they're faithful to that aqueous data point that you got, and you can translate it onto the array easier. These are more like nano test tubes. These are 0.2 nanoliter volume, 100 micrometers, and pretty much do any kind of biochemical test. We've shown that you can look at genetics markers, protein markers, metabolites, small molecules. We've shown that drug levels, we've shown that drugs going into somebody and different protein markers are going down with drug treatment. So you can also load these with reagents, and we found that to be come in very handy as we're trying to make this easier to use. The big step that we have taken is to put this into a microfluidic flow cell. So we have our own flow cell here, and you're looking at about 220 spots on that, and it now allows us to run PCR in the same chamber as the array. So it's a closed amplicon approach. We have a valveless design, and we also can do immunoassays with it. The big step that we took as a company is that we're now printing on plastic film, all plastic. So now we're talking about pennies for materials costs versus the $8 I have to pay for a really good piece of glass, let alone a silicon wafer. And then this enables reel-to-reel manufacturing, which is used in the immunochromatographic strips, and that's the vision of the company, is to be able to print these arrays in a flow cell in a reel-to-reel production line. And just to our last piece of the puzzle, I'll be finished, is the true tip, which is able to, our own technology that is able to process samples, large samples, raw sputum. We have various data from the users in the crowd here that are showing that this sample preparation is working very well compared to magnetic beads or spin columns. And it's actually shown to be very good as a front-end technology for next-gen sequencing platforms. So here's our platform. We have the sample prep workstation, our 2000 detection system, and our 3000, which is fully integrated. And here's our long list of collaborators. Those who are underlined is where we have systems deployed, and the rest are funding or very helpful collaborators that we have. So thank you.
All right. Thank you very much. Chuck, would you mind staying up here?